Hello everyone. Today let's understand cytokines. Cytokines are really important molecules. And you have seen I've created these dots over there, and these are not just dots, they are protein molecules. Okay? They are protein molecules and they are soluble molecules. That means they are present in our blood. They are not present on any cellular membrane. And usually they have glycomolecules or the sugar molecules. That's why they are known as glycoproteins. And they are very, very important for our immune system, our immune cells. Cytokines, they play a very important role in intercellular signaling pathway. Means communication between the cells. For example, you can have one cell right over here. Let me create that. You have one cell which is leukocyte, let's say, and then you have other cell like macrophage. There is a communication between these two. Right, that communication which leads to the immune response is governed with the cytokine signaling pathway. That's why cytokines are really important. Cytokines, they play an important role between two important immune system, innate immune system and adaptive immune system. So they are the connecting bridge. And cytokines, they play a very, very important role in various infections, including viruses. So that is what I'm creating over here, viruses. Second one, bacterial infections. So whether it is bacterial infection, whether it is virus infection, and whether it is a damage caused by a toxin, bacterial toxin, you have cytokines playing important role. Plus parasitic infections. If there is worm present in the body, right? Even in case of cancer, when there is uncontrolled growth of cells, right? That's a tumor. So I'm just showing you a lot of these cells, and that is the cancerous cell or cancer cellular mass. So cytokines plays very important role in those things. Next is, let's talk about basic terms that we use in cytokine biology. The first one is monokines. What are monokines? Monokines, the cytokines released by specific cells called monocytes as well as macrophages. So I'm just using that, phage sign. Next is lymphokines. As the name indicates, lymphocytes producing cytokines are the lymphokines, specifically T cells. T cell produce cytokines, they are called lymphokines. Interleukins, they are ILs. Interleukins are produced by leukocytes, right? So let me write it down. Leukocytes, when leukocytes, they produce some cytokines and then they will also affect the leukocytes. Those leukocytes producing those molecules are called interleukins. So those molecules are interleukins. Next is, they are important for immune cell growth, important for differentiation of immune cells, as well as activation of the immune cell for performing a particular function. And then you have another category which is colony stimulating factors. Colony stimulating factors, what are those, right? So colony stimulating factors, specifically you have, let's say, blood precursors. Blood precursors are there, and those molecules, CSF molecules, they play an important role in growth as well as differentiation of those blood precursors. And that happens in case of inside the bone marrow, where the process of haematopoiesis is going on. Right over there, those molecules are the colony stimulating factors. Now let's talk about the chemokines. Chemokines are really important for our immune system, and they play a very important role in movement of the immune cells. So moving cells from one part to another part is governed by the chemokines. For example, you have, let's say, bacteria present on a particular site, which is site of infection, and then these chemokines are going to attract these cells, immune cells, to the site of the infection. That blue color cell you are seeing is the leukocyte. Leukocytes, they are going to come to the site of the infection right over here. That is going to govern by chemokines. Okay. And that process is known as chemotaxis as well as chemokinesis. Chemokinesis, that is governed by these chemokines. Example for this is RANTES, Rentis. And then next example is MIPS. Okay. MIPS. MIPS, let me write it down. MIPS, macrophage inflammatory protein. And these are central mediators. MIPS specifically is the central mediator of your inflammation. So if you want to maybe quantify inflammation, you can look for MIP protein quantification. Now let's talk about haematopoietins. Haematopoietins, they are important for blood cell production. If we need to increase the blood cell production, you have these molecules. So HSC cell stimulation is going to be governed by these molecules, plus the formation of RBCs, erythropoietic processes. You have the example is erythropoietin, that is going to govern the production of RBCs. And then you have further colony stimulating factors that we have already covered, important for haematopoiesis. These are some of the important molecules that play an important role in case of, the functioning and the control of the cytokine, as well as we have understood the basic terminology of what kind of cytokines are there, what are their specific role. Okay, now let's move on to the next part. All right, now let's talk about interleukins. What are interleukins? Interleukins are really important for growth of the immune cells, for activation, as well as differentiation of the immune cells. There are various cells that are present in our body, specifically immune cells. They are being, the growth is being regulated, differentiation as well as activation. Let's suppose we have this particular cell, and it's going to communicate with another cell. And one side you have lymphocytes, another side you have leukocytes. The communication is happening because of the interleukins, right? Example is IL-1, interleukin 1, interleukin 18. These are really important interleukins. We'll talk about the function later on. Now, some of the other molecules, including tumor necrosis factor family. There are family of the protein molecules that are really important for tumor necrosis, right? So TNF alpha is known as the name of the molecule, important for inflammation, fever, and then immune regulation is very, very important function of TNF alpha family molecules. It also triggers apoptosis. Apoptosis is nothing but when cell is going to die itself, it's going to form those apoptotic bodies, and then without the potent inflammatory response, it's going to die. And that's very important process. Example is you have TNF alpha, that is another protein. You have TNF beta, okay, TNF beta, and then a fast ligand protein. Okay, now let's talk about the mode of cytokine action. How the cytokine actually perform their task. The first mechanism is autocrine action. Cytokines, they have autocrine action. Autocrine action means the cell, and I think let me create the illustration. This is the cell. It's going to produce the cytokine, and this will also act on the same cytokine backwards. So the same cell is going to get influenced by this. Next is paracrine. Paracrine, you have a cell. It's going to produce the cytokine, but it's going to affect a different cell in the body. That's the paracrine. But nearby, the cells that are present nearby is the paracrine mode of action. The example, if we talk about example of for autocrine, where the cell is getting affected, example is T cell. T cell usually do that. Okay, and example is interleukin 2. In case of your paracrine, you have macrophages that are going to affect the immune cells. So the cytokines, they are getting produced from macrophages. Next is endocrine action. Endocrine action, let's understand what is that. Endocrine action. Endocrine action means cell is going to produce the cytokines. They are going to enter into circulation. This is the blood vessel, and they are going to move to a distance very, very far, let's say, cell, and it's going to affect the function of those particular cells. So after entering into circulation, distant cells, they are going to get affected. For example, let's say liver cells, right? Liver cells, they are getting affected with the cytokines, and the cell which is producing, immune cell producing IL-6, and that is going to target the liver cell. So you can imagine how important it is. The example is liver cell is going to produce the acute phase protein by the activation of IL-6. Very, very important regulation of these molecules. So the induction is going to happen. Why? Because of the virus infection again, because of the bacterial infection, because of the parasitic infection. All those things are going to produce these cytokines, and then cancerous cells also are going to induce that. So inflammation, another process, as well as T cell activation is going to cause the release of these cytokines. So basically what happens is there is one cytokine that is one family is going to released by one cell, and then that cell is going to again release the cytokine. It's going to affect another cell. Another cytokine is going to increase the production, and then you have a network of these cytokines coming from different cells. So a particular channel of the molecular activation is there. Talk about cytokine signaling. Very important process. Cytokine signaling. What happens in cytokine signaling? In cytokine signaling, cell usually have a receptor on its surface. This is the receptor, and then cytokine, this is the protein molecule, is going to bind to that particular receptor, and binding with very high affinity is going to happen. The affinity is very strong of these cytokines to their receptors. That's why the important point is, even the lowest concentration, even the low concentration of these molecules is going to induce very strong response inside the cell, right? So now let's talk about how the activation happens. So you have a receptor present on the cell surface. This is surface, this is receptor, and then cytokine came, it got interacted with this receptor, binding happened. Now intercellular signaling pathway activated further proteins that will cause the activation of the gene expression. Now gene is going to get activated, and those are going to produce further protein molecules, and that is the effector molecules. These are because of the expression of various genes. So you have expression profile that is coming, and then further what is going to happen, that you have a cell, right? Whenever this interaction happens, let me draw it again. It's going to bind. It's going to do various things. The number one is it's going to produce another set of cytokines. Second one, it's going to activate adhesion molecules. So let me write it down. Adhesion molecules. So adhesion, but the process of attachment of the cell is going to happen. Next is proteases are also induced by the activation of cytokines. Further, lipid mediators. Let me create the structure of lipid molecules. Lipid mediators are also activated by the cytokines. Next is nitric oxide synthase is also known to be produced. Let me write oxide synthase is also known to be produced by action of cytokines. So why this is happening? Because high antimicrobial action is required, because you have a lot of bacteria, a lot of virus, so they should be killed because of the action of these molecules. So killing is the final outcome of the cytokine action. What is happening as cell is getting activated, there is a receptor present on the cell, cytokine will bind to that particular receptor, and then further signaling will happen, ultimately producing molecules that can actually kill cells, viruses, bacteria. Cells means some cancerous cells that should be killed. So basically this is going to regulate the important process, the effector process. That is why cytokines are really important. All right, let's talk about another molecular family, which is interferons. We'll talk about the different types of interferons. So now, whenever you have a cell, specifically immune cell, is getting infected with virus. Just imagine virus is inside this particular cell. So what is going to happen? That it's going to produce those molecules called interferon, and specifically interferon alpha. And that interferon alpha is going to activate the T cell, right? And then you have NK cells. So usually activated T cells and NK cells, they're going to release these kind of molecules. And further, it has antiviral activity induction. You have MSC molecules that are getting expressed, specifically class 1 MSC molecules. They're going to expressed in a larger quantity. Next, as you can see over here, MSC class 1. Okay. Next is interferon beta. So let's imagine you have a cell, specifically the cell called fibroblasts. These fibroblasts, if they get virus infected, again, virus is inside, and if there is virus infection, they're going to produce another class molecules called interferon beta. And then antivirus action is going to happen. So just because you have fibroblasts involved, you have interferon beta. Next is interferon gamma. So you have three interferons, alpha, beta, and gamma. And this is produced by Th1 cells. So just imagine this is T cell, specifically Th1 cell, and that is, is going to affect the functioning of cytotoxic T cells. So it's really important for the functioning of cytotoxic T cells as well as NK cells. So this is going to get released by activated macrophages. Next is T cells. Next is NK cells, and it's going to enhance the MSC class 1 as well as MSC class 2 expression. Both of these molecules are going to be expressed, plus antigen presentation is going to enhance. So you can imagine why these things are happening, because there is virus infection. Now we will be discussing individual molecules. Like, let's discuss the IL-1. IL-1 is produced by monocytes, macrophages. We all need to understand and remember what are the cells: endothelial cells as well as fibroblast. These are the cells that produces IL-1, and it is major inflammatory mediator. It's pro-inflammatory molecule. It's also fever-inducing molecule, so it's called endogenous pyrogens, right? So this is also known as endogenous pyrogens. Next is it also activates T cells as well as B cells, and because of that, it's going to boost our immune system. Immunity is going to enhance. Now let's talk about IL-2, and this is again a protein produced by Th1 cells, and its role is it's T cell growth factor. It's going to be involved in proliferation of T cells. So proliferation of T cell is going to happen. You have very, very high NK cell activity because of IL-2, very, very high beta cell activation because of the IL-2, and also antibody production because B cell activation is going to happen. So high antibody production. Now let's move to another interleukin 3. Interleukin 3, again protein molecule, and you have particular cells called T cells producing, and also the mast cells producing IL-3. Okay, it is going to stimulate the production of granulocytes. That's very, very important function of this cytokine. Production of granulocyte is going to happen. Haematopoiesis, or also macrophages production is going to happen. The process of haematopoiesis, let me write it down. Haematopoiesis is going to happen because of this IL-3. IL-4, Th2 cells, mast cells, and basophils. They are going to produce IL-4, and it's going to affect the Th2 cell differentiation and also B cell activation. Let me create that B cell right over here. B cell activation is going to happen, and because of that, antibodies are going to get produced. Look at these antibodies. Antibody production is going to increase, enhance. Next is IL-5. So many interleukins. IL-5, Th2 cells produced by Th2 cells. So it's going to activate eosinophils, and very important for parasitic infections. Further, B cell activity is going to enhance. That's why antibodies are going to produce in very, very high amount, right? Now you understand that what are interferons and also what are interleukins, especially 1, 2, 3, 4, and 5. That is what we have covered. Let's move on to the next part. All right, let's discuss IL-6. IL-6 is produced by Th2 cells. Th2 cells, macrophages, let me write it down, macrophages are also going to produce IL-6. Fibroblasts, very, very important cell line, that is going to produce IL-6. The function is, it's going to activate liver to produce acute phase proteins. And further, hematopoietic activity is going to happen because of this. IL-6, T cell plus B cell growth is also regulated by IL-6. Next is IL-8. So many interleukins. IL-8. Monocytes. Okay. Next, endothelial cells, specifically endothelial cells and fibroblasts. They are able to produce IL-8, right? And this is very, very important molecule. Please remember the function of this. This is IL-8, and it's going to attract very, very important cell, very, very important cell for our innate immunity, which is neutrophil. Neutrophils are going to get attracted because of IL-8. You need to remember that. So migration of neutrophil is going to happen. Now let's talk about IL-10. They are going to get produced by Th2 cells, B cells, macrophages, right? And look at that. This particular interleukin is anti-inflammatory in nature, opposite to most of the interleukins we have studied. Anti-inflammatory is going to, downregulate the functioning of other interleukins. So interleukin IL-1 is going to go down. IL-6 is going to go down. Next is TNF alpha as well as interferon gamma is going to go down because of this IL-10 anti-inflammatory molecule. And this is why, because excessive immune system is being controlled by this cytokine. Next, let's talk about TNF alpha. TNF alpha produced by various immune cells, including macrophages. Second one, T cells. Okay. Next one, NK cells. These cells, they are able to produce TNF alpha, inflammatory cytokine. Whenever there is inflammation, TNF alpha is involved. Whenever there is fever, acute phase protein production you have TNF alpha that is playing very, very important role. It is also toxic to tumor cells. TNF alpha is toxic to tumor cells. Now let's talk about TNF beta. It's also known as lymphotoxin. Now we have T cells that are involved in this case. You have B cells that are involved in this case, and it's going to produce TNF alpha, which is inflammatory in nature, right? Okay, now let's talk about another important molecule, which is G-CSF, G colony stimulating factor, produced by T cells, produced by macrophages, produced by neutrophils, and then the role is to produce more and more neutrophils. Let's write it down. More neutrophils are going to get produced. Differentiation as well as activation of the neutrophil is going to happen because of the G colony stimulating factor. That we need to understand. So neutrophils are the target for G-CSF. All right, now let's talk about M-CSF. Colony stimulating factor M. T cells, macrophages, and fibroblasts. They're going to produce this particular molecule. The role is the macrophages, these particular macrophages, the growth or differentiation of the macrophages is going to happen because of the M-CSF. Okay, so this is going to regulate the macrophage function. Let me write it down. Macrophage function is regulated by this. Okay, so I think we have covered a lot of different cytokines. Let's understand what is the overall role of these cytokines, right? Cytokines, they are playing very, very important role. If we put cytokines in the circle in the center, they are important for immune cell function. Okay, they are also important for attracting immune cells to the site of infection. They're important for inducing fever, also reducing fevers. They are important for inflammation because they can balance it out. So low inflammatory response as well as high inflammatory response is regulated by this. Cytokines, specifically cytokine network, is very, very important for growth and activation of the immune cell. Right? That is what we have covered so far. So cytokines in very much detail. I've tried to lay out all those important cytokines that are present and understanding the basic innate as well as adaptive immunity. You need to create your own notes and study in detail about those cytokines that we have covered. They are very, very important when it comes to exams, when it comes to specifically competitive exams like NET. You need to understand what are the cytokines, what are their roles, and how they are linked with different conditions and all. So that is very, very important. All right, now let's meet in the next video where we will be understanding more interesting topic and really studying those topics in depth. That is the motto of the biology goal and research goal, right? This particular video is also included as the part of the course. So I recommend go to the research goal website, complete that particular course, because all those important videos that are important for this particular topic are categorized in the course format in research goal. On YouTube video, you are able to just see these videos isolated somewhere, and you are not going to make sense that which video I should be watching first. And that's why I've created that structured course for your competitive exam preparation, for your research understanding. Everything is there on research goal. Link is there in the description box. Please check it out. All right, let's meet in the next video.